Okay, so I apologise for the abrupt ending to the previous video. I didn't foresee the fire alarm going off. Okay, right, so we're in the process of discussing the blood-brain barrier. Okay, so we were discussing the structure of a normal capillary. So normal capillaries, and it's a little bit difficult to show here, so I'm just going to draw another picture before I go on to explain this. So let's draw a cross-section of a capillary. Okay, so if we draw a cross-section of a capillary, the structure of a capillary might look a little bit like this. Here is an endothelial cell here lining the capillary, okay, and then underneath the endothelial cell there is then another layer, and this is what I can't really show you on that other picture, which is the basement membrane. Okay, so even where there are these holes, it is not as though there is an actual full hole there. There is going to be basement membrane over here, but there are portions where there are no endothelial cells lining the wall of the capillary. Okay, so where the only portion that actually is there is the basement membrane. Okay, so this is the basement membrane, here BM, and this is the endothelial cell, which I'll abbreviate to EC. Okay, so in normal capillaries elsewhere in the body then, you have these leaky capillaries where certain portions of the capillary wall is literally just the basement membrane, which is a web of proteins, okay? And that makes these capillaries quite leaky, and it means that many things that are in the blood can easily get into the interstitial fluid, okay? However, in the brain, it's different. In the brain, there are no holes in the endothelial cells. The endothelial cells are everywhere, okay? So always a capillary wall is made up of an endothelial cell and then a basement membrane underneath it. So if I put these in, the endothelial cells don't have gaps in between them, so there are more endothelial cells basically. Okay, and these endothelial cells will have tight junctions in between them. Okay, so this means that these capillaries in the brain are much less leaky than the capillaries elsewhere in the body. Moreover, it means that if anything wants to move from the lumen of the capillary into the interstitial fluid, it has to go through an endothelial cell. So let's draw this here. So here's one endothelial cell, here's a neighbouring endothelial cell. We have tight junctions in between the neighbouring endothelial cells here. Okay, if something wants to move into the brain, it needs to go across the endothelial cells. Okay, now the apical membrane of the endothelial cells, which is the membrane facing into the lumen of the blood vessels, has P-glycoproteins on it. Okay, and again, it's the same case as was for the epithelial cells in the gastrointestinal tract, where you have the two different portions of the membrane of these endothelial cells. You have an apical portion which faces into the lumen, okay, and then you have a basolateral portion which faces onto the basement membrane and onto the interstitial fluid of the tissue, okay, which is in this case the brain. And the P-glycoproteins are reserved for the apical portion of membrane. So let's say here is one. Okay, so if you have got drug molecules, these toxic molecules into the bloodstream, then we can protect the brain at least from them, because to get into the brain they have to first go into the endothelial cells, and then in the endothelial cells they will be pumped back out into the lumen by the P-glycoproteins. Okay, so here is how P-glycoprotein protects the brain from toxic hydrophobic molecules such as drug molecules. Okay, and this is how it influences pharmacokinetics, because it's influencing distribution here. So this is how it influences distribution in pharmacokinetics, because in this case P-glycoprotein stops drug molecules getting into the brain, certain drug molecules getting into the brain, because it will stop them crossing the blood-brain barrier by pumping them back into the blood whenever they move into the endothelial cells of the capillaries in the brain. Okay, right, so that's how P-glycoprotein influences distribution. Now what we want to turn our attention to is how it influences elimination of the drug. Okay, so the other portion of pharmacokinetics that I said P-glycoprotein has a role in. Okay, so elimination involves the liver and the kidneys. So let's start by discussing the liver. Okay, so the liver is a massive, massive great organ, which basically consists of loads of tubes that are all eventually draining into the bile duct. Okay, so let me draw one of these. So we're going to take a cross-section through one of these tubes. Okay, and we're going to see the cells that line the tube here. Okay, like so. So this is the tube here, and then we've got the cells that are lining the tube here. Okay, like so. Now, the tube has a special name. It's what is known as a bile canaliculus. Okay, so this is a bile canaliculus, and bile is going to be being made by these cells that line the bile canaliculus, and it will be moving through the bile canaliculus, and eventually it will drain through many other tubes into the bile duct. Okay, right. So these cells then that line the bile canaliculus, these are what are known as the hepatocytes, the main cells of the liver. Okay, and the hepatocytes are epithelial cells once again, so they have two portions of their membrane that are separated by tight junctions in between the hepatocytes. Okay, now the portion of membrane that faces into the lumen of the bile canaliculus is known as the apical portion of the membrane, and the portion that faces out from the bile canaliculus is known as the basolateral portion. Okay, so this is the basolateral portion. Now the basolateral portion of the membrane is going to be exposed to blood vessels that are coming through the liver. Okay, specifically a special type of capillary known as sinusoids, okay, which are very, very large, very leaky capillaries. Okay, so again, these capillaries will have points where there are no endothelial cells making up the wall, and it's just basement membrane. Okay, so I'll draw this here. So I'll start by putting the basement membrane here. Okay, like so. So here's the basement membrane. And it's a very large capillary this time, larger than normal capillaries. Okay, here is an endothelial cell here, and then we might have a gap between this endothelial cell and the next endothelial cell over here, and then maybe I'll put one down here as well. Okay, so these are the endothelial cells here, and this capillary here is known as a sinusoid. Okay, right. So what can now happen is if the liver wants to remove drugs from the blood, then what it can do is the drugs can come in from the blood into the hepatocytes here, and then the hepatocytes can excrete them into the bile. Okay, now there are two different ways that this can happen. Okay, if the drug is a very hydrophobic molecule, then it can just go into the hepatocyte easily. Okay, it can just diffuse through the hepatocyte membrane into the cytoplasm of the hepatocyte, and then the hepatocyte has P-glycoproteins on the apical side of its membrane, which I'll put here, which can then move that hydrophobic drug molecule into the bile, and then it can be excreted with the bile into the intestine and hopefully out into the external world. Okay, so that's if the drug molecule is hydrophobic, it can just be excreted by P-glycoprotein in this way into the bile. Okay, if the drug molecule is not hydrophobic, however, what then has to happen is it has to be processed to make it hydrophobic. Okay, so hepatocytes are capable of processing drugs. Okay, so the processing of drugs, the metabolism of drugs, is divided up into two types of reactions. Okay, phase 1 reactions and then phase 2 reactions. Okay, so in phase 1 reactions, what happens is the drug molecule is changed so that you make new functional groups. Okay, so phase 1 reactions are often known as functionalization. Okay, and you make new functional groups such as alcohol groups, thiol groups, and amino groups. Okay, and these are groups that will then be used to react with other molecules in phase 2 reactions. Okay, so phase 2 reactions are also called conjugation reactions, and in phase 2 conjugation reactions, what happens is other molecules are now attached onto a drug molecule by means of the functional groups that you've now added onto that. Okay, so you react other molecules with the functional groups, and those new molecules end up attached to your drug molecule. Okay, so molecules that can be conjugated onto drug molecules include glutathione. Okay, in addition, methyl groups can be added on. Okay, acetyl groups can be added on. and also glucuronic acid can be added on. Okay, glucuronic acid. Okay, and adding these groups on then make the molecule more hydrophobic, and then what can happen is that new conjugated drug molecule can then be excreted by P-glycoprotein into the bile, and therefore it can be excreted from the body by the liver. Okay, so that's the role of P-glycoprotein in the excretion of drug molecules by the liver. I also would like to mention how it's involved in the excretion of drug molecules by the kidney. Okay, so let's just go over a little bit of basic renal anatomy and physiology, because generally people aren't too too comfortable with the anatomy and physiology of the kidney. So we'll just go over a bit of basic kidney physiology. Okay, so I'm drawing now the right kidney here. Okay, here is the ureter coming out here, the right ureter. So this is the ureter. And then the kidney is divided up into two main portions. Okay, there is what's known as the cortex, which I'm drawing now, with these trabeculae which extend down into the more deeper depths here. Okay, like so. So this portion of the kidney here is known as the renal cortex. Okay, then you have these trabeculae, which are effectively extensions of the cortex downwards into the centre. So these are known as trabeculae. Okay, and then these other portions, which I'll colour in red here, which are in between neighbouring trabeculae here. Okay, and these are what are known as the renal medulla portions. Specifically, each one of these little red structures that I'm showing here is known as a renal pyramid. Okay, right. So these are the renal pyramids, and they're the main portion of the renal medulla. Okay, so renal pyramids, and they're in the medulla, which means the middle portion of the kidney. Okay, right. And then this basin here, which is where all the urine drains into before draining into the ureter rather, is known as the renal pelvis. Okay, so that's the sort of gross anatomy of the kidney. Now let's have a look at where nephrons are within this structure. Okay, so basically a nephron looks like this. These are the different components of a nephron. So you start off with what's known as Bowman's capsule here, and then the first portion of the nephron is known as the proximal convoluted tubule, and is very convoluted, like so. Then you have a portion known as the loop of Henle, and then you have another portion called the distal convoluted tubule, which then drains into a collecting duct. Okay, so here is the loop of Henle, then we have the distal convoluted tubule, and then the thing will drain into a collecting duct which will have loads of other nephrons draining into it as well. Okay, right, so let's label up the different portions here. So this first portion here, Bowman's capsule. Okay, this portion is known as Bowman's capsule. Okay, now this is going to be the point where fluid from the blood is filtered into this space here within the Bowman's capsule. Okay, and that's going to be the starting point for making urine. Okay, so let's just put this here. So you'll have an afferent arteriole coming in here, an efferent arteriole coming out here, and then you'll have loads of these little capillaries in the middle here, and this cluster of capillaries is known as the glomerulus. Okay, and what happens is fluid from the capillaries is squeezed out into the Bowman's space here, the Bowman's capsule. Okay, so I'll highlight this up. So this portion here, this is Bowman's capsule here. Okay, and this is where the first fluid that's going to be the precursor to urine is going to be accumulating. Okay, so this special collection of capillaries here, this is what's known as a glomerulus, a renal glomerulus. Okay, right, so fluid is squeezed out from these capillaries into the Bowman's capsule, and then what's going to happen is it's going to make its way through this portion here, which is known as the proximal convoluted tubule. So for lack of space, I've just abbreviated that down to PCT. Then it's going to go through this portion here, which is known as the loop of Henle. Okay, And then after the loop of Henle, you have the distal convoluted tubule, the DCT, which will then drain into a collecting duct here. Okay, right. So the basic idea then is that this fluid is going to move through all of these tubes, and then throughout these tubes what's going to be happening is essential nutrients that are within this fluid are going to be reabsorbed. So for instance, electrolytes are going to be reabsorbed, amino acids are going to be reabsorbed, glucose and other carbohydrates are going to be reabsorbed. Okay, and then horrible things that you want to get rid of, such as urea, they're going to stay within the fluid, and they're going to eventually go into the collecting duct and then drain into the pelvis to be excreted as urine. Okay, so if I just draw the picture of a nephron onto my bigger picture here, I'm just wondering where to try and put it. Here is Bowman's capsule in the cortex. The proximal convoluted tubule is also in the cortex. The loop of Henle dips down into the renal pelvis like so. Then the distal convoluted tubule is also in the pelvis, not in the pelvis, in the cortex, and then the collecting duct will make its way through the renal pyramid and then drain into the pelvis down here. Okay, so that's where the nephron sort of fits into this larger picture of the anatomy of the kidney. Okay, right. So, basically, the cells that line these renal tubules, okay, and the name for the combination of all of these different tubules, they're called renal tubules, basically. So this word refers to not just one section of this, but all sections of this. So the proximal convoluted tubule, the loop of Henle, the distal convoluted tubule, they're all counted as renal tubules. Okay, so the cells of these renal tubules are going to have, again, an apical face and a basolateral face. So let's just draw this. So let's draw one of these renal tubule cells here. So again, it's an epithelial cell, and it will have tight junctions between itself and its neighbours. Okay, so there'll be a tight junction here, which splits the membrane into the apical portion, which is the portion actually lining the lumen of whichever renal tubule you're talking about, whether it be the proximal convoluted tubule, the loop of Henle, or the distal convoluted tubule, or even the collecting duct. Okay, and then you've got the basolateral surface, which is then in contact with the blood. Okay, so remember, these cells are involved in reabsorbing all sorts of valuable nutrients. Now, how are they going to return these nutrients into the blood? Well, basically, they're going to take them from the lumen, they'll go into the cytoplasm for a while, and then they'll move them into the blood on the basolateral side. Okay? Right, now there are certain drug molecules that you do not want to be reabsorbed. Okay, if you want to eliminate drug molecules from the body, you don't want them to be reabsorbed. Okay, and P-glycoprotein is therefore found on the apical surface of these renal tubule cells, and its job is to secrete drug molecules back out into the lumen to make sure that they're not being reabsorbed. In addition, what can actually happen is drug molecules can move from the bloodstream into the renal tubule cells and then be secreted into the lumen. So the renal tubule cells can actually move drug molecules from the blood into the contents within the lumen so that they are excreted within the within the urine. Okay, so this is how P-glycoprotein can play a very important role in the elimination of drug molecules through the kidney. Okay, right, so that's P-glycoprotein's physiological role in pharmacokinetics explored a little bit more. What I want to go into now is, before we talk about a very interesting correlate of P-glycoprotein being so involved in pharmacokinetics, what I want to discuss is a few other ABC multi-drug transporters that are found in humans, okay, because many of these have very, very similar functions to P-glycoprotein. So the first one that I want to mention is known as ABCG2. Okay, so this is the ABC transporter. So again, this naming system tells you instantly that this is an ABC transporter in humans. Okay, G2 then says that it's family G and it's family member 2. Okay, so ABCG2. Okay, now ABCG2 is a half transporter. Okay, so remember, it's going to have to dimerize in order to actually produce a functional active transporter. So if I just remind you of this picture here, it will have one transmembrane domain here, which will consist of that cluster of 6 membrane-spanning alpha helices. Then it will have its nucleotide binding domain here. Okay, and therefore it will have to form a homodimer to be functional. Okay, now pretty much it's identical to P-glycoprotein in its distribution and its function. Okay, so all of the places that we've just discussed P-glycoprotein being involved, you can also find ABCG2. So for instance, on the intestinal cells, and where have I put that picture? Here we go. On the intestinal cells, you can also find ABCG2 here as well. Okay, performing the same role as P-glycoprotein, which is extruding these potentially toxic hydrophobic molecules back into the lumen of the gastrointestinal tract. Okay, on the blood-brain barrier, you can also find ABCG2, also performing this role of protecting the brain from potentially toxic hydrophobic molecules. Okay, on the apical membrane of the hepatocytes, you can also find ABCG2 playing this role of excreting hydrophobic molecules into the bile. And then again, on renal tubule cells, you can also find ABCG2 playing the role of excreting hydrophobic molecules into the urine. Okay, right, so it performs many of the same functions as P-glycoprotein. You can also find it upregulated in multidrug resistant cancer cells, okay, and this is the origin of its other name. So ABCG2 has another name. It's also known as BCRP. Okay, now what does this stand for? This stands for breast, that's the B, cancer, that's the C, and then resistance protein, that's the RP. So breast cancer resistance protein. So again, it can be found upregulated in cancer cells, and it can extrude a very similar collection of drugs to P-glycoprotein, basically. Okay, so there's another ABC multi-drug transporter in humans. Okay, I'd like to talk about 2 more examples. Okay, and these 2 are quite interesting because they actually have structures that are different from the normal structures that we've seen. So the normal structures that we've seen are the full ABC transporters and the half ABC transporters. But these final 2 that I want to discuss, which are ABCC2 and also ABCC1, and you might be wondering, well, why has he done it in this bizarre order? Okay, but I am going to talk about ABCC2 before I talk about ABCC1. Okay, so these 2, which are ABC transporters in humans in the C family, these actually have a very different structure to the full transporters and the half transporters. They are full transporters, but they don't have that same structure that we've looked at before. In fact, they have an additional cluster of 5 membrane-spanning alpha helices. Let me just show you this. So if this is the membrane here, let's say this is the extracellular fluid side of the membrane here, and then this is the cytoplasmic side of the membrane here. Okay, so they actually have their amino terminus extracellularly, not intracellularly. Then they have a first cluster of 5 membrane-spanning alpha helices, and then it's exactly the same as the full transporter. So then they have the cluster of 6 membrane-spanning alpha helices, followed by a nucleotide binding domain, then another cluster of 6 membrane-spanning alpha helices here, followed by a nucleotide binding domain here, and then finally followed by the C-terminal here. Okay, so let me label up these portions. So this first cluster of 5 membrane-spanning alpha helices, this is then known as transmembrane domain 0. Okay, so this portion here, which I'll colour in red, this is TMD0. Okay, and then everything's exactly the same as the full transporter. Okay, so here, this is transmembrane domain 1. Okay, then we have transmembrane domain 2 here, okay, which I'll colour in blue as well, and then the 2 nucleotide binding domains. Okay, so again, these function as full transporters. The individual protein on its own is enough to actively transport substrate molecules out of the cell. Okay, but they do actually have a slightly different structure to the full transporters, so I thought that was worth mentioning. Okay, right. So we'll start with what ABCC2 actually does then. So ABCC2 also has another name. It's also known as MRP2, so you might see it referred to as that. And this refers to multidrug, that's the M, and then resistance associated is the R. So resistance and then dash associated, and then protein 2. So this is the multidrug resistance associated protein 2. Okay, now, its function in physiology and in pharmacokinetics is similar to P-glycoprotein, but not quite as broad as P-glycoprotein. It's found on the renal cells and the hepatic cells, and it's involved in drug elimination. So again, on the apical surface of renal cells here, you could have ABCC2 also performing this function. Okay, so in reality you'll have a mixture of P-glycoprotein, ABCG2 and ABCC2 all performing these same functions on the apical surface of renal tubular cells. In addition, on the bile canaliculi, the apical surface of these hepatocytes, you can also find ABCC2 there, and it's again involved in excreting these hydrophobic molecules into the bile. Okay, right, finally, ABCC1. This is quite different from the other three that we've talked about. This is also known as MRP1, okay, multidrug resistance associated protein 1, okay. This one is actually found on all cells of the human body, okay, and effectively it's a personal vacuum cleaner for every single cell in your body, okay. It's a way of getting rid of potentially dangerous hydrophobic molecules, okay, and again it is heavily associated with multi-drug resistance in cancer cells. So cancer cells can acquire mutations that lead to upregulation of this, and it can then lead to multi-drug resistance in these cancer cells because ABCC1 extrudes such a multitude of different hydrophobic compounds, including many drugs. Okay, right. So those are the 4 ABC MDR transporters that I want to mention in humans then. Okay, one little clinical correlate that I want to talk about now is the potential then for modulators of these. Okay, so it was very exciting initially when P-glycoprotein was found to be the cause of multi-drug resistance in certain cancer cells, because we thought we could just create drugs that would inhibit it, and that would then make our drugs effective again. Okay, now there are drugs that are capable of inhibiting P-glycoprotein. So an example of a drug that was developed for exactly this purpose of inhibiting P-glycoprotein and therefore reversing resistance in certain cancer cells is a drug known as tariquidar. Okay, now of course the inherent problem with giving a P-glycoprotein inhibitor is that P-glycoprotein has so many important physiological functions. And to illustrate the problem with giving P-glycoprotein inhibitors, I'm going to give you a very nice example of drug interactions. Okay, so the example I'm going to give is what happens if you take verapamil, okay, which is a P-glycoprotein inhibitor, with, for instance, a drug called Loperamide, which is an anti-diarrheal drug that you can actually buy over the counter in the UK. Okay, so let me explain. Verapamil is actually a P-glycoprotein inhibitor. That's not the reason it's used clinically. The reason it's used clinically is because it's a voltage-gated calcium channel blocker, okay, and it's used to treat hypertension. Okay, however, it is a very effective P-glycoprotein inhibitor. Okay, so if you're taking verapamil for your hypertension and you then go to a chemist and buy some loperamide as an anti-diarrheal medication, this can have a very bad interaction basically. So let me explain how loperamide works, and then we'll talk about what happens if you take these two drugs together. Okay, so loperamide is actually a mu-opioid receptor agonist. Now that little brief description of this drug is actually the same brief description that you would give for the drug morphine. Okay, however, why can this drug be given over the counter as an anti-diarrheal drug? Well, basically the answer is that it cannot get into the brain. Now, before I go further with that, let me just explain how loperamide is used as an anti-diarrheal drug. So one of the famous side effects of opiates is that they cause constipation. Okay, now the reason they cause constipation is that they inhibit acetylcholine neurons that are innervating smooth muscle cells surrounding the gastrointestinal tract. So if I just draw a little picture here of the gastrointestinal tract. So basically, if this is a portion of the gastrointestinal tract, one of the key linings of the gastrointestinal tract is a muscle layer. Okay, you have a lot of smooth muscle cells surrounding the gastrointestinal tract, and these need to contract in order to constrict the lumen and push food forward, so they're involved in peristalsis. Okay, now, they are innervated, okay, by acetylcholine neurons. Okay, so this is a neuron which will be releasing acetylcholine onto the smooth muscle cell here. So this is the smooth muscle cell, SMC. Okay, and the acetylcholine will then be activating muscarinic acetylcholine receptors, which will then lead to contraction in the smooth muscle cell, and that's how you get peristalsis and the movement of food through the bowel. Okay, now, these acetylcholine neurons here, they actually have receptors on their axon terminals here, which are called mu-opioid receptors. Okay, so OR here is for opioid receptor. Okay, now, certain drugs can activate these mu-opioid receptors. Notably, morphine can activate mu-opioid receptors, but loperamide is also an agonist of these receptors. And when loperamide activates these receptors on these acetylcholine neurons, what it does is it stops these axon terminals from releasing as much acetylcholine, so it hugely reduces the amount of acetylcholine that these axon terminals will secrete, basically. Okay, and that then leads to less contraction of the smooth muscle cells and therefore less rapid movement of food through the intestine, and that's the basis of how loperamide can be used as an anti-diarrhea drug. Okay, so this is an anti-diarrhea drug. Okay, and it's available over the counter. Okay, so you don't need to go to a doctor to be prescribed loperamide. Okay, now, the reason that loperamide can be given and not have the effects that morphine has on the brain is that loperamide does not cross the blood-brain barrier, whereas morphine, which is also a mu-opioid receptor agonist, does cross the blood-brain barrier. Now, if loperamide was to cross the blood-brain barrier, then it would have a similar effect to morphine. Okay, but it doesn't. Now, why doesn't it cross the blood-brain barrier? Well, the reason it doesn't cross the blood-brain barrier, as you probably suspected here, is that it is pumped out of these endothelial cells by P-glycoprotein. So if you take loperamide at the same time as taking verapamil, a P-glycoprotein inhibitor, then loperamide is no longer pumped out of these endothelial cells by the P-glycoprotein, and it does get into the brain, and it will trigger an effect similar to morphine. Okay, and that is what is known as a drug interaction. Okay, so when this drug on its own does not cause the effects of morphine on the brain, but when you take it with another drug, it does. Okay, so that's a clinically relevant drug interaction. Okay, and there are loads of other examples of drug interactions. Okay, particularly verapamil, because of its effects on P-glycoprotein, hugely changes the pharmacokinetics of many drugs, and that can have serious consequences. Okay, right. We'll call it there for this video, and in the next video what we'll do is we'll move on to the next family of multi-drug transporters that I want to discuss, which is the RND transporters.